Really delighted to have Ann Cerruti present our keynote presentation today. Dr. Cerruti received her MD and PhD from Emory University School of Medicine and completed her general pediatrics residency at the Children's Hospital of Philadelphia. She returned to Emory for subspecialty fellowship training in pediatric infectious disease, and then after that came onto the faculty. We certainly are proud of her for being one of our star K-12 awardees, and as a really a model for career development. Dr. Cerruti is now Associate Professor of Pediatrics and Infectious Disease. She's the Associate Division Chief for Basic and Translational Research in the Division of Infectious Disease and Director of the Center for Childhood Infections and Vaccines. She's an attending physician on the ID Consult Service at Children's Healthcare of Atlanta, and the Medical Director of the Ponce Family and Youth Clinic, part of the infectious disease program of the Grady Health System. Her research focuses on finding a cure for HIV using non-primate models. Her lab aims are to identify cellular and anatomic HIV reservoirs that represent the key obstacles to an HIV cure. Dr. Cerruti is extraordinarily dedicated to career development of early career emerging scientists all along the career pathway. She's on the faculty of the Immunology and Molecular Pathogenesis graduate training program of the Laney Graduate School. She's Associate Director for Clinical Affairs of the MD PhD program at Emory, and she is the founding director of the Pediatric Residency Investigative Scholars at Emory, or PRIZE program, which supports research in our residency training programs. So at this time, it's a pleasure to present Dr. Ann Cerruti speaking on the topic of my path as a pediatric physician scientist, the tortoise and the hare. Ann, take it away.
Let me add one more word to that introduction, that very elegant introduction. Dr. Ann Cerruti, as Dr. Joyner said, is a role model, and I hope that all of you young people attending this virtual seminar find a moment to either meet with her, talk with her, and be inspired by what Ann has been able to achieve as a former fellow of our program just a few years ago to become a nationally renowned scientist, and we continue to find time to be a mentor for young people. That is pretty remarkable. So, Ann, thank you for everything you do. We are forever grateful to you.
Hi, thank you very much. Can you hear me okay? Okay, great. Well, thanks very much to Lucky and Clint, as well as Stacy, for inviting me to give this talk today. It is a true honor. I'm going to share my screen. Okay. So can you see that now? Oh, okay, hold on. I think I better go back to the... Okay, so you can see the full view of the slide, correct?
Yes, it looks good.
Okay, perfect. All right. So thank you so much for this opportunity and this invitation. This is definitely a new kind of talk for me. I was asked to sort of talk about my path and my career training, and not to specifically focus on my science. So I had over the past few days a lot of imposter syndrome kicking in as I tried to think about what I could tell you that would be helpful in terms of career development. So hopefully I will be able to kind of pull out some pearls that can be useful to some of you on this Zoom call or Zoom talk. I think when I reflected on my career path, there were some things that kind of stood out to me. And I chose the theme of the tortoise and the hare to describe them to you. So as you all know and heard, I pursued a combined MD PhD, and that's often considered to be a tortoise way of going about training because it's quite long. But as I think I'll tell you and kind of elaborate on the introduction that Clint gave, my path to getting from kind of a young undergraduate, kind of just starting out and being interested in science to where I am now was filled with a lot of different fits and starts. And some of those were intentional pauses that had to do with life choices that I made. And so I sort of wanted to share those with you. And then on the flip side of that, because of some of those pauses, there are other times in my career where I tried to kind of jumpstart and go as fast as I could to sort of keep the momentum going. So as we go through the slides, I put a little tortoise in the corner where we're talking about a pause or maybe something slightly deviating from the kind of quote unquote typical path. And then a hare on the slides where I tried to kind of move a bit quicker. And as we go through the talk, I thought about sort of what I could tell you that was sort of broader than just talking about, you know, my particular path. And it's not meant to in any way be a prescription for what I think is the right thing for other people to do, but rather just to provide one example about how training can go and how it can lead to success or lack of success in various aspects down the road. And the pearls that I think in terms of career development that I wanted to convey and that I do often say, I think, to the MD PhD students that I interact with are here. And so the first is not to wait for training to end to start your life. The second is to be open to opportunity. And the third is to cultivate your physician scientist identity. And this last one is something I'm really passionate about, and it speaks to what Lucky mentioned recently, and sorry, Clint as well, in terms of the Pediatric Residency Investigative Scholars Program. So I'll describe that in a little bit more detail at the end of the slides. So to start with, the first point, don't wait for training to end to start your life. For me, this had to do with starting a family. But I think it's more broadly applicable to many other aspects of life. People have many different hobbies and passions and advocacy issues that they want to address. And so I'll talk you through kind of my path and how my family grew over the course of my training. But I just want to say that, you know, for people who have another, you know, choose a different route, I think some of the concepts are still broadly applicable. So this is what we'll go through over the next few slides. So this is sort of the career timeline going from starting undergraduate in 1992 to the present day in 2020. And it involves all of the different classical training steps that a physician scientist embarks upon that you can see on this timeline here. But as I mentioned before, there are several different pauses or turtle moments, and then several different hare moments as well. And this training did involve a lot of travel, kind of traversing the United States from the east to the west and then the northeast to the south. And I think that's really been an important part for me to be able to learn from and engage with people from all over the country. And frankly, science is engaging with people from all over the world. And so that for me has been sort of one of the things I found most rewarding about being in a scientific career. And I just want to say, kind of as an aside, this travel that I'm showing you back and forth across the United States, while, you know, it was sort of exciting for me at the time, it is also associated with some hardship at various times. But I also want to point out that it in no way compares to the large number of people who are in the US now who had to leave their country of origin, either by choice or by necessity, to come and train and work in the United States. And so I don't want to, you know, underestimate what that has entailed, and I've lived with it in terms of the experiences that my husband has. So I will talk you through more of that as well. Okay, so just getting started to paint a picture of where this all began for me. And so I think, you know, all of us who have a passion in their research careers, it all came from somewhere. And I'm often asked why I study HIV and why that has been the thing that I've dedicated both my clinical and my research life to. So here's some timelines and some key events that were happening in the world that I grew up in, and probably some of the other folks on this call grew up in as well. And so in my, you know, what I would call formative adolescent years, basically HIV and AIDS came upon us and hit the national headlines, and was something that was, you know, frankly quite frightening. You can see in 1992, that's the year I started college, that AIDS became the #1 cause of death in the United States for men ages 25 to 44. There were a lot of sort of brochures in the mail that were sent home, understanding AIDS. You asked for questions about AIDS. And, you know, many folks who were sort of living that time as practicing physicians have incredible stories to tell about kind of how their practice of medicine changed. I was not at that stage of my career yet, but it still had a very profound impact on the choices that I made going forward. And in the top right panel is the first AIDS memorial quilt that has a square of the quilt for each person who passed away from HIV and AIDS. And this was on the National Mall in 1997. And I just wanted to show also on the bottom right, the bottom right picture is a digitized image of this quilt that was made in 2012. And interestingly, as I was kind of putting these slides together, I saw that in 1992, you know, one of the international AIDS conferences that we attend pretty much every year as scientists was moved out of the United States due to immigration restrictions on people living with HIV and AIDS. And those immigration restrictions were lifted by the Obama administration in 2012. And so that's why the International AIDS Conference was held in Washington, D.C. And this digitized image of the quilt was made kind of in honor of that event. So I went off to college, very passionate about HIV, and I studied biology and got very interested in immunology as well. And as a junior, I found a lab to work in. And this is pictured as Dr. Gail Skowron, who was a physician scientist herself and had a research lab. And I actually found, kind of in scouring my files this past weekend, my honors thesis from Brown, where I looked at HIV-specific CTL precursors found in infected persons. So this was kind of super exciting for me, and I saw what it was like to be a practicing physician scientist. And so I left college thinking, okay, I want to do this. I want to go to medical school, or I want to go to graduate school, but I wasn't really sure exactly what the path was, and I didn't know much about MD, PhD. I didn't have any family members who were doctors or scientists, so it was a little bit something that I needed to take more time to figure out what I wanted to do and how I wanted to do it. So here's the first tortoise that you see in the left corner. Leaving college, you know, as I said, I was interested in medicine and science. But frankly, at the age of 21 and having lived in the Northeast for my entire life, my main goal was to live in California. And so I basically drove to California with a friend and set up shop in San Francisco, and then started looking for jobs. I don't think that would really be too possible given the apartment rents these days, but it was possible at that time. And so I eventually found a position at Chiron Corporation, which was a biotech firm in Emeryville in the East Bay, so across from San Francisco. And I was hired to work in the adjuvant research group. And I was working on an influenza vaccine that was adjuvanted with something called MF59. And so, you know, what this sort of pause was a sort of a life desire to live in California. And what it meant was that I kind of took a deviation from my passion of studying HIV. I still thought that that's what I wanted to do, but I thought that this deviation was worth it. Now, unfortunately, I had a crash course in what it can be like to work in industry. And this company, Chiron, had a failed herpes vaccine that was part of a different group that I didn't work in. But as a result of that, there were a number of layoffs, of which I was one of them. And so that was a kind of a wake-up call for me. So at that point, I started investigating other jobs in academia again, and not in the biotech industry. And I interviewed at a number of different places, and I thought, okay, well, let me get back into HIV for sure, because that's really where my passions lie. But what I ended up doing actually was something completely different. And so I found a job as a research specialist at the Rockefeller University, working in Ralph Steinman's lab on dendritic cells and studying EBV, in fact. And so the reason for this, 2-year pause as well in terms of the standard training was the environment that I experienced at Rockefeller University, and just the amazing science that was happening, the international postdocs that filled the lab. And it really was I think I kind of knew it was a unique opportunity, but I didn't really know. And over the time being there, it became clear that this was really a great kind of model for how to run a lab and conduct science. And, there's many people who have gone on from that lab, whether they were graduate students or postdocs, to do kind of amazing science. And so experiencing that was really unique. And I also wanted to point out, I didn't know this obviously at the time, but Ralph Steinman, if you don't know, went on to win the Nobel Prize for the discovery of dendritic cells. Unfortunately, it had to be posthumously awarded to him. But again, that was a pretty amazing experience. So during my time, both at Chiron and at Rockefeller, I made it a point to interview all the scientists I encountered, whether they had trained as a medical doctor or a PhD or both in some cases, and really kind of poll the room and see sort of what the recommendations were, what the advantages and disadvantages were. And for me, here having all those conversations and kind of seeing where my passions were, I decided to pursue an MD PhD program. And so I applied many places. I interviewed at a few. I was accepted at even fewer, but one of which was Emory. And so I'm showing here on the left, for those that haven't been down there, the Emory Vaccine Center and the Yerkes National Primate Research Center. And this Emory Vaccine Center was just being built when I came for my interviews and starting here at Emory. And there were a nice group of HIV scientists who are really dedicated to finding an HIV vaccine that was being put together there by Rafi Ahmed. And so that was really kind of what, in my mind, brought me to Emory, was to do that type of science. Again, HIV being the passion and the thread that kind of leads through all of these different experiences. However, as I was going through my files this weekend to try to find that thesis from Brown, I came across some papers that I had saved for who knows what reason, but this is my list of pros and cons for the Emory University MD PhD program that I made when I was obviously trying to decide whether I should come here or not. So some of the things that I've underlined, I think, were probably pretty appropriate and still ring true today. Grady Hospital is obviously a wonderful place to train as a medical student at that time. And I think even now it's a growing and exciting time at Emory in Atlanta. The support is excellent. And I think that still rings true kind of across Emory. But there were a couple of other things I thought were quite interesting about my pros and cons list. So, one thing is second on my list in the pro was that they had a very nice athletic center. And on my con was that they had a not a great cafeteria. So I think what this tells you is that, you know, you lead with your mind in terms of the science, but you also have to listen to, you know, and be honest with yourself about what things are important to you at that given time in your life. And so at that time I was a very avid athlete and runner, and so the athletic center was important to me, and I guess I was also an avid eater, so the cafeteria was equally as important. And just as a sign of the times, I thought it was quite funny to see that in the cons I would need a car and maybe even a computer. So that was pretty exciting, and I'll stop dating myself now. So during the course of my time at Emory, there were a number of formative events in my life. The first of which is depicted here. This is the first paper that I published as a graduate student, and it stemmed from work that I did in a rotation in the lab that I eventually joined with Mark Feinberg, another physician scientist. But I'm highlighting the person and showing a picture of the person that I have collaborated with many times since then, and also married as well. And so this kind of unique moment of meeting somebody that you're going to spend the rest of your life with and scientifically collaborate with was really quite exciting. And one thing that we like to point out is that we didn't know each other when this paper was published, but we met very soon thereafter. So I'm just showing here kind of to keep the thread and the theme of what science I pursued over these years going. So these are the papers that I published during my PhD. And you can see on the right, there was some HIV specific work, and this was from a rotation that I did in Janet McNichol's lab when I was in the Immunology and Molecular Pathogenesis graduate program. But as you can see on the left, the majority of work that I did in my PhD was related to pox viruses and host pathogen interactions between pox viruses and the immune system. And the reason for this was because it was some preclinical work to try to lay the foundation for an HIV vaccine. And I was very excited about it at the time, but it still shows that there was some bit of deviation from this path of wanting to work on something that will, directly affect the lives of people living with HIV. I put a hair up in the corner there because although most of the time you wouldn't expect that an MD PhD would be considered to be something fast, which it isn't. At that time, it was possible to finish in 7 years. It's not really possible to do that anymore just based on the clinical requirements, but I was determined to do so. And so I still think of that as a bit of a hair time in my life and my training. So the other important things that happened during the course of my MD PhD at Emory is that the birth of my daughter and my first son in 2004 and 2006. And so these things are obviously life-changing and necessitated a bit of another moment of a pause in my life. And so, when I finished the MD PhD program, I decided to, quote unquote, take a year off with a newborn and a 2-year-old. And so I spent most of my time pushing them around in a double stroller that's depicted on the left, although the one that I had was way less fancy than this one at that time. And I was applying to residency programs, during this period. And I also did have the opportunity to do a mini postdoc. And this is the first time that I really started to work with non-human primate samples, which has become sort of the cornerstone of what I do now. Even though I did my PhD at the Emory Vaccine Center, which is attached to the Yerkes National Primate Research Center, I actually didn't do any non-human primate work at that time. So this was kind of a pause, but it enabled me to learn about non-human primate research in a way that really, I think, shaped my future ability to do work in that field. So the next phase, kind of powering through here, was residency at the Children's Hospital of Philadelphia. And this phase for me involved basically no research at all. And that's something that, it'll come up a little bit later, is a bit of a danger zone for physician scientists in training. And it's a time in what you'll see as the leaky pipeline that there is significant attrition. For me, I would say that it was a choice that I made to try to get through the clinical training and really focus on learning how to be a doctor as kind of quickly and straightforwardly as possible, because and I think I was able to do that because I was living with somebody who I collaborated with in the past and was had an active research lab. So I think I was able to kind of, in a very unique situation where I was able to stay abreast of where the science was going while still pursuing my full-time clinical training. And then the final big event that I'll talk about was the birth of my second son in 2010. And so this is what necessitated the next pause, in my view. So when we returned to Atlanta and Emory, I didn't go straight from residency program into the fellowship program, but took a 1-year, quote unquote break, if you're calling these things in the typical training path. And I actually came back to my academic home and did some research with my husband, Guido Silvestri, at the Yerkes National Primate Research Center. But I also established myself as a physician at the Grady Infectious Diseases Program, where we care for HIV-infected children and adolescents. And so that's something that from 2010 on, I've worked consistently in that practice, and it's really been a kind of key component of my career. Then jumping ahead into fellowship, I was able to do that in an abbreviated fashion. So did a fellowship in 2 years rather than 3, and continued in the area of HIV, mostly focusing on pathogenesis work and transmission. So again, kind of getting me closer and closer to what my high school self wanted to do, but still not quite working on the direct translational work that is able to kind of have the potential to go straight ahead into clinical trials for people living with HIV. So at the end of all this, I get to say that I'm 39 years old and have my own lab. This is in 2013. And what I think, as I've conveyed to you over the course of this talk, is that you can take a number of different deviations from the path, but everything sort of, I think when you have time to reflect, everything builds upon each other and brings you to the point. And so the exciting thing, as I think I've alluded to, when you have your own lab, you are able to make your own decisions with the wise help of the grant reviewers that are involved in the process, and do the work that you wanted to do all along since high school. And so what we do in my lab now is translational studies, trying to understand HIV persistence, both in infants and adults. But we also test translational approaches towards curing HIV. And this is really kind of, as I have said numerous times, this is really what I wanted to do all along. And so it's really exciting to be able to kind of make that happen at this point in my career. And so the other thing that has been really exciting over the past few years is being able to work with a great group of individuals, both in the kind of research arena, but also within the infectious diseases division, within the Grady Infectious Diseases program that I talked to you before about, and in the Emory MD PhD program. And so I just love all of these pictures on this slide, and I've been very fortunate to be able to work with all of these people and kind of work together to continue to publish and to make, sort of contribute to the scientific field of HIV. So moving on to the next pearl. So I kind of went through that timeline of my life to talk to you about, how life happens when training happens. And then as I was reflecting on the things that, since I've become a faculty member, I think what has sort of changed my trajectory and maybe kind of jump-started some things in some cases was an openness to opportunity. And so let's talk a little bit about that now. One issue that I think has come up recently is this kind of issue of how to say no. And so, as I just mentioned, I want to talk to you about being open to opportunity, which seems to be the opposite of this, how to say no and just say no, the art of no, all of these headlines that you see. I think that this sort of being in vogue in how to decline opportunities has been kind of an unfortunate consequence of some of the wellness programs that have rolled out that all too often focus on changing ourselves and not changing the system. And so all of these sort of advice columns that you see and a lot of, I think, talks that we hear are about sort of how to change yourself to establish your goals. And of course, it's not going to be all cut and dry. There will be, sometimes when you need to say no and sometimes when you can't say yes. But I think that saying yes really provides a lot of opportunities and benefits. So this is these are, different things, saying yes to reviewing a paper or reviewing grants, serving on a committee, teaching a class, collaborating with someone new, exploring a new scientific field. All of these things, I think, can have benefits in our career, particularly as junior faculty members. So saying yes can open doors, new doors and new avenues of collaboration. It engages our curiosity. It promotes continued learning and growth. It can foster deeper connections with other people, with your institution, with your community, and our sense of giving back. And it also demonstrates a confidence in your abilities. So when you say yes to something, you're saying, yes, I can do that. And I think all of these things really can be very helpful to kind of grow as a junior faculty and even, as you become a senior faculty member as well. So just some examples from my own recent life. So I said yes to working on a clinical trial that I had never envisioned myself being a part of before. So learning how to write a protocol and conduct a clinical trial. I said yes to kind of exploring research options with 2 new viruses that one I had never heard of when I was a new faculty member, and 2 didn't exist when I was a new faculty member. So these are Zika virus and SARS-CoV-2. And then also working on a committee to plan a meeting that the Pediatric Academic Society's meeting that really allowed me to interact with people from multiple different fields of study, people that I knew in residency and only had sort of come back to interact with as a result of this meeting. And it just sort of opens your eyes to other points of view, but also you can see the parallels in how things work in different fields. And then also shown here is the Emory and Children's Center for Childhood Infections and Vaccines that I was asked to lead. And that was something that I didn't know a whole lot about when I started, but it's really become a big part of my career and an opportunity to advance the field of infectious diseases within Emory and Children's. So the last thing I'll talk about is cultivating a physician-scientist identity. And this really does apply primarily to physicians. So I apologize if there are non-physicians listening in. But it's been a really big part of kind of my understanding of what I am doing and why I'm doing it. And sort of, I think the professional identity formation has really been something that is really critical to sustainability within this career. And I don't want to imply that it's specific to those who've done an MD and a PhD training. It's something that applies to any physician who is choosing to pursue science. So what is professional identity? It can be defined as a set of values, beliefs, affiliations, aspirations that all synchronize with the norms of a particular profession. And there's been, I think, a lot of kind of research and emphasis on this within medical education, less so within sort of the dual identities of a physician or clinician and a scientist. And this is an article that I was pointed to by Joanna Bonsall, who is one of the co-directors of the Emory-MD PhD program, that really sort of talks through some suggestions for how to strengthen the professional identity formation within the training all throughout both clinical and science training. So I just wanted to highlight a couple of things that we've been thinking about as we're thinking about the prize program that was mentioned earlier. So I think everyone on this call likely possesses all of these personal characteristics that are shown here in the box. These are factors that support the development of your health professional identity. But then there's other things in terms of program design that are highlighted with the arrows that can really support the integration of these dual features. So both the clinician aspect of our careers and the scientist aspect of our careers. And so these involve a structured research experience, the socialization process, facilitating reconciliation of your personal and dual professional identities, as well as training in team-based research and collaboration. That's really what science is these days. And so it's really critical that there's training in that process. So why is this all important? Why do we care? One of the things that there's been a lot of data and statistics on over the past few years is the leaky pipeline of clinician scientists and physician scientists. And so, as I mentioned, these are people entering the postgraduate pipeline either as MD PhDs or as MDs with an interest in research. And so there is a risk for attrition going into residency and fellowship, and then that period in time when maybe you're neither a fellow nor a faculty, you're in a kind of the initial stages of your post-fellowship career. And so what ends up happening in some cases is that people who have a passion for science don't end up pursuing it. And this is really critical in pediatrics because we need child health researchers. You all gave amazing talks this morning, and I anticipate there'll be even more this afternoon. And it's really critical that that work that we're all doing translates into improving the health of children. And this leaky pipeline continues in the faculty period. And in particular, it can be difficult or can be sort of more pronounced for different groups of individuals, including women, as well as people who tend to be underrepresented in science and medicine. And so I just pulled out a couple of kind of what I think are sort of the three key issues that unless we address them, we're not really going to make, a lot of progress in this area. And so the first is shown on the top left, and this is an article that I saw just very recently, and I pulled out the information about pediatrics, and this refers to the gender pay gap, and it shows as the percentage of pediatricians has increased, so that's the line that's going up, as the percentage of pediatricians has increased since the mid-70s to 2017, their overall proportion of the salaries for physicians has declined. And this is true in a couple of other kind of female-focused, what have become female-focused disciplines. Another thing that has been pointed out, and this is from a New England Journal of Medicine paper from last year, is that women and underrepresented folks are still excluded from leadership position, the highest level of leadership position. So here this is department chairs and medical school deans. And I don't know if you can see this line going all the way out, but if the current rate continues, the end of this line goes until 2070 or around 2066 for it to be at a 50-50 equity rate between males and females. And so this paper was calling for a call for term limits in academic medicine to try to rectify the situation. And then the final piece that I think is obvious and yet still unsolved is the lack of universal healthcare, or not healthcare, I'm sorry, that's a problem too, but universal childcare. And that really can be for women, but also for families and fathers as well, can be a true impediment to being able to pursue the careers that people want to pursue. And this is, of course, being exacerbated by the current coronavirus pandemic. So to me, these are really three key threats that need to be addressed. Oops. Okay, so I sort of have outlined what the problem is. And one solution that we are pursuing here refers to that leaky pipeline during the clinical training. So this doesn't really address the issues I just raised in terms of the faculty years. But we're really excited about this new program, the Pediatric Residency Investigative Scholars at Emory. I was so excited to hear the research presented by the residents earlier today. This is a program that is supported by the Department of Pediatrics and Lucky Jane, and it is also being conducted in collaboration with the residency program, and Susie Buchter has been an excellent champion of this as well. I'm working with Dr. Stacy Heilman as well as Jennifer Villasenor to kind of implement this work. And just to kind of briefly plug it to this group, our goal is to develop high caliber pediatrician scientists with the potential for long-term impact on child health. And one of the key pieces of this is that we are focusing on professional identity formation as a pediatrician scientist. So as I referred to in the past couple of slides, this has been shown to be a critical piece to kind of success and sustainability in a future career, in which there often is sort of this dualism in terms of one demands maybe different from the other. And our program is targeted for MD PhD as well as MD residents, and it's kind of one of our stresses is that it's highly flexible. So there's a lot of ways that it can be approached. And really, I'll just say that this is a way to formalize a lot of the research efforts that have already been going on within the residency program in the Department of Pediatrics, as well as to provide career development sessions. And some of the faculty on this call will likely be called upon as we start these programs. If you are willing to give a session to the residents, we will take you up on that. And so a lot of this is related to what their research project will be, but also how can we support their career development as physician scientists. So just to summarize, the three career development pearls that I thought I could share with you are the first, don't wait for training to end to start your life. So this is really about who we are. The second, be open to opportunity. To me, I think this is about how we grow and how we advance within our careers. And then the third is this cultivating a physician scientist identity. And I think really what this is a reminder of why we do this, which is sometimes a hard career to do, and there's a lot of ups and downs and rejections and things of that nature. And so we do this because we care about the patients that we treat, and we're also passionate about the science that we're pursuing. So thank you to everybody who's on the call and for the invitation again, Lucky and Clint. I think that, I just wanted to share also kind of where those babies ended up. This is a recent picture. And so thanks for your attention. I think there's time for questions, so I'm happy to take any that we have.
Absolutely, and that was just phenomenal. I'm giving you my applause if you can't see my little emoji here. I'm inviting people to use the chat feature to enter your questions, but I actually have a question that was prompted by one of the slides that you showed about the characteristics that exemplify someone who will be successful. I think it was, like you said, related to MD PhD program, but it really is really generalizable to researchers in general. And so I hope this isn't too philosophical for you, but one of the characteristics was resilience, and another was the tolerance for complexity and ambiguity. And my question for you is, there are, I think, some people in life who naturally have that, but I wanted to ask your opinion on if this is something that could be learned by someone who maybe isn't a natural in those areas, and if so, what would your recommendation be?
That's a really good question. I think that, you know, I don't pretend to be an expert kind of in that particular area about, you know, how you would train in that regard. But I do think that the tolerance for ambiguity is a part of, certainly a part of scientific training, but I think a part of medical training as well. You know, we all get to the point in our training where, you know, maybe particularly in ID, I don't know if that's just because I know, but you're reading the textbook for an answer to a particular patient question, and it says, okay, in this particular case, you'd want to consult an ID expert, you know, and you then realize, well, I'm the ID expert and I'm trying to read what to do in this particular textbook. So I think, you know, we get to that point, we know that there are limits to our knowledge in medicine and science. And so I think that if you don't have a tolerance for ambiguity, that, you know, you may have weeded yourself out at a certain point in time, and not have gotten to kind of the point where you are. Resilience, I think there's been a lot of, you know, discussion about resilience over the past few years, and I think that, you know, maybe some of it has to do with early life events, and sort of challenges that we've all faced. But again, I don't profess to be sort of an expert in that area. I think that may be something that's just a kind of not innate to your genetics, but sort of relates to your upbringing.
Yeah, and I do one. Yes.
Hey, Stacy, this is Lucky. Do you mind if I ask a question?
Please go ahead.
Anne, thank you so much for this wonderful talk. I'm sure there are going to be a lot of questions that you have generated in people's minds. But one that comes to me perpetually is the fact that with the longevity that has been given to us by science, by medicine, by everything else that has happened in the past several decades, a year or two of slowing down here and there, like you have done and so elegantly explained, doesn't really seem to have impacted your career. Whereas I can tell you that when I was in the same stages of my career as you were, it wasn't acceptable to just take a year off. And that is my biggest regret, that when we had our two children, I was back at work a week later. So can you tell us if you have any regrets about the times when you slowed down dramatically, and would you have done things any differently?
Yeah, thanks for that question. I think, you know, at the time, it did feel scary to do that, and it felt potentially, you know, disastrous. But at the same time, because, you know, it really, as you say, it's not typically what is done. I think a lot of people do take time off before they start their medical school or scientific training. But sort of those steps kind of within once you hit that stride, it's not typically done. I can say I have no regrets at all. And it just was, it was necessary. It was sort of the right thing to do for myself and for my family. And so it was not even really a question. And so we just made it work. And I think that that is something that I do want to convey to people, is that, you know, that's sort of what I meant by just living your life. You know, you, in retrospect, looking back, those 1-2 years here and there, you know, they flew by in an instant, and I don't think that they, you know, really impacted me negatively. They probably impacted me positively in terms of kind of being able to, when I was in a particular training program, to you know, give it as my 100%.
Thank you.
to piggyback on that, Holly Bowser-Heaton has a question about how do you enjoy the slower times? Like, how do you really know you're in the slower times and take pause and don't get stressed about it? Do you have any tips for, especially during this COVID, during the quarantine and the research slowdown?
Yeah, thanks for that question, Holly. You're asking the tough one. I think, you know, so it's hard to sort of think sort of back retrospectively, and you know, I think we should also say that, you know, having a newborn and a 2-year-old is not slow by any means, right? So I think that there are different ways to kind of interpret it, but I think in just kind of as a more general response, you know, I think that we all would love to say that when we go home at night, you know, we put the phone away and the computer away and we don't do any work. And I just think that's not our reality, unfortunately. I do think when my kids were younger, I did more of that, and I felt like I was able to, you know, turn off and on. And actually, in some ways, having kids during clinical training sort of enabled that more. And now that they're older, I'm more able to kind of use my time efficiently with them. They don't usually want to hang out with me much anymore anyway. But so I think, you know, again, like I said, I'm not saying what I did as a prescription for anybody else by any means, but I think hopefully it's just an example of how to, you know, think about negotiating your life, you know, with your profession and your personal life.
Thank you. Jessica Raper has a question. You mentioned taking opportunities which have broadened your research into other viruses, but how do you take the opportunities while also avoiding being characterized as lacking focus?
That's a good question, Jessica. And Jessica is one of the people that I've worked with very closely on Zika virus. So I consider that to be a great opportunity that we took together. I think that you have to build a narrative for yourself. You know, I think kind of at the most basic level on your CV, when you're writing your 50-word, I think it is, research focus, you know, it's pretty hard to kind of, if you are going in multiple different directions, to kind of get that to show that you are focused. But I think if you build a narrative that, you know, for me, initially it was very HIV focused, but now it is, you know, studying virologic disease, you know, virus infections in terms of their relationship with the human immune system, and sort of you can find a way to sort of circumscribe what it is that you do into one broader focus. I think, you know, if you're talking about, you know, doing behavioral neuroscience part of the time and then doing, you know, BSL-3 virology another part of the time, maybe that's a little bit harder to reconcile. But I think for most of us, the opportunities that do present themselves are, you know, within a general field of, you know, focus and scope. And so it just is about kind of how you incorporate that into, you know, what you put forth in your elevator pitch in terms of what you're working on.
Yeah, I like that answer. It's really all about, can you justify it? Can you tell the story? Can you put it together in such a way that it makes sense and is complementary and synergistic? That's some great advice. I also want to ask you about your Twitter handle. So I'm a faithful follower at Ann Trudy on Twitter. And tell us a little bit about how you use that outlet in your personal, professional, and as an outlet, and how it supports your scientific career.
Okay, so I got into Twitter now probably a couple years ago, I think, and it was really because some of the MD PhD students at Emory had kind of started to, well, they were probably using it for a long time before I started, but they had a kind of a master class in how to use Twitter at a meeting that I went to, and so I decided to try it. And I think you go through, at least in my experience and what I've heard from other people, you go through various stages of being a Twitter user. First, you're kind of a lurker, so you look and see what other people are posting, and you look and see who other people follow, and then you kind of learn to kind of who is posting things that you're interested in. I by no means feel like I'm an expert. There's a lot of other super users on the Emory campus that I could point you to. And I think at first, you know, I think a lot of it is used to kind of promote people around you, and particularly students, but also, you know, other folks within, you know, CCIV. I like to post their papers or comment. And then, you know, I think there is some degree of self-promotion that happens as well, and that's a part of our lives as scientists that I think we all should probably acknowledge. But then really the other flip side of it is information. And I think it's particularly during the COVID pandemic, you know, where I see and learn about papers is from Twitter first. And you see and hear about them immediately. And so it's really a way to stay abreast of the scientific literature. I think there's another kind of side to it that is a bit of kind of gets hyper political, and it's a lot of kind of everybody repeating the same thing. And so I try to stay out of that as much as possible. But I think you can really, for me, it has been a way to kind of stay abreast of the literature and to make connections with people that I wouldn't normally interact with on a regular basis.
Thank you. So another question I'm wondering about is, you talked a lot about, and you articulated oh so well about what your goals were at each level of your career stage. What are your goals now? What would you be mad at yourself for not accomplishing before you retire?
Well, as I mentioned, I sort of feel like I only just got started, and I'm not thinking about retiring too soon, hopefully. But I think, you know, right now I really am focused on the science that we are doing, and it's a really exciting time, you know, in infectious diseases, you know, of course, the COVID pandemic, but in our HIV work, we have a number of studies, translational studies in non-human primates that are going on now that I really think have the potential to contribute to, you know, where we go next with a clinical cure for HIV infected patients. And so that's really the driver. I think from a kind of career trajectory, you know, that some of that is still things that are percolating in my mind. And I think, as I mentioned, I like the idea of saying yes to many opportunities, but I think that, you know, now's the time to deliver on them. And so you have to kind of get to a point where you do have to call a little bit. And, you know, I think that I have found that I do like to kind of the individualized mentoring. And so I really want to dedicate, you know, a lot of my time to that going forward. And then we'll just kind of see where things go.
Okay, thank you. Are there any questions any final questions? I think I just want to say on a personal note, and how we've been working together a little bit more closely since PRIZE and with a couple of other projects. And it's just so wonderful working closely with you. You're just such a smart and savvy, decisive individual, a great communicator, and truly altruistic. And I think you've exemplified that with your talk today, and just showing everyone kind of what makes you tick and some best practices as we navigate this thing called life and science. So I hope everyone will join me in virtually applauding Dr. Trudy for all the great work she's done and for sharing some of her best pearls of wisdom with us. So thank you.
Thank you.
Okay. Right. I think we're a little bit ahead of schedule. I'm going to go ahead and just go over, think we might have a sort of a different group of people here this afternoon for the basic science part of the presentation. And so assuming that that's correct, and assuming that we're ready to go and we can kind of give you a little bit of an overview of what to expect the rest of today, I just want to mention a few things. One is we ask that everyone keep muted during the program. Barbara Kilborn is our sort of Oz in this point, and she's got all the controls. So if you forget and are, you know, walking around your house or something, or your dog is barking, she'll mute on your behalf. We're going to be using, for the afternoon session, we have a number of judges who'll be scoring and evaluating the oral presentations. And at the end, we'll be giving a prize for the best oral presentation, most highly ranked, as well as the most highly ranked poster. And so the judges will be allowed to speak their questions and ask, and everyone else, we just ask you to use chat, and I'll be moderating at the end. After the presentations, we'll have about 3 minutes per person to ask those questions. Just real quick on the program, if my computer is cooperating right now, there's a variety of links that hopefully you all have seen. Let's see here. My apologies. There we go. Okay, so I'm just going to share my screen with you for a moment, just to orient you to all of the materials that you need. Okay, so if you go to pedsresearch.org and on the homepage, you can get to the Pediatric Research and Career Development Symposium. Really, here's all the materials that you're going to need for today, but I'm going to focus on the program detailed agenda, which is a booklet that has everything you need to know. I'm actually going to turn off my video because I'm having, let me try this instead. Okay, we're going to go here. I'm having some internet issues today, which is not great for an all-day career symposium. But at any rate, here we are about halfway down in the program. So we just had our wonderful keynote presentation by our very own Ann Trudy. So now what we're going to do is we're going to have 2 basic science oral presentations here and here. Like I said, each of them will be followed by about 3 minutes of Q&A. Then we're going to have a short break. Then right after the break, we're going to do this thing that we're calling a poster session blitz. And what that is, it's each of the 6 poster presenters are going to give you just a 3-minute vignette of their research and what data and conclusions are on their posters. It's just enough to whet your appetite. They're not going to tell you the whole story, but what it's designed to do is give you just a little taste of what they're going to present. And then later in the presentation, we'll have a virtual poster session. So more on that in just a second. After that, we'll come back and we'll have 2 more oral presentations. And then this is where the virtual fun begins with the basic science poster Zoom rooms. You can see here in the presentation, each of them have their own Zoom room links. So if you listen to Holly Bowser-Heaton talk about her great science and you want to know more, you have questions, all you do is click on this link. It takes you to her Zoom room, and it can just be like you were in the foyer of HSRB, where you can talk to her and ask questions and have more of that one-on-one interaction. So that's for networking and more scientific purposes. Then we'll come back at 4 o'clock when we'll announce the best basic science oral presentation and the poster PDF winner. And after that, we'll do the wrap up. So with that, we're right on time at 1:30. So I am going to stop my share and we will turn it over to Clint Joyner, who is going to be our moderator for this afternoon's session.
Okay, let me turn on my video so you can see me. So our first basic science presenter is Dr. Navdeep Ajita, who is a postdoctoral fellow in the Division of Rheumatology and Pediatrics. Navdeep has been with Emory for 2.5 years, having completed her residency at Emory, and she's currently pursuing additional postdoctoral training here in immunology. Her mentor is Dr. Dmitry Shakhmedov, and her research focus is immune responses to TLR signaling. So take it away, Navdeep.
Thank you, Dr. Jointer, and thank you, Dr. Chowdhury, for your inspirational talk. I've been set up to introduce the afternoon basic science talk, so here we go. I am talking about vaccines and how they modulate our immune response. We use vaccines for a vast variety of pathogens that we encounter, such as influenza, COVID, and a whole spectrum of other bacterial and viral pathogens. However, our current vaccine model, which is our live attenuated vaccine, historically is our best method of vaccine, but it is very immunogenic and can create many side effects. In that regard, we are studying subunit vaccines, which contain fragments of the pathogen in order to stimulate the same immune response in a safer and less immunogenic fashion. In our studies, we have studied, or we are studying, pathogen-like recognition receptors, PRRs, which are toll-like receptors. These toll-like receptors can be found on our front line of immune defense, such as macrophages and dendritic cells. These cells can sense and pick up particles from PAMPs, which are pathogen-associated molecular pathogens that are part of bacteria and viruses that attack us every day. These allow us to study the immune response that our first line of defense or innate immunity can host and promote. Next slide, please. So in this regard, we synthesized pathogen-like particles in a microparticle or a nanoparticle format to stimulate the pathogens we are stimulating our immune response. We have co-loaded these pathogens with adjuvants that are similar and stimulate these TLRs that we're studying. Here we have a model of how we did our pathogen-like particle. Our particle, macroparticles or nanoparticles, have been double emulsed. So the first emulsification enloads PEI, which will have a negative charge on the molecule, and thus when we co-load, it'll have a positive charge, and so when we co-load our adjuvants, they will hydrophobically load as a negative charge. Next slide, please. Here, we are studying different adjuvants and TLR receptors that are being stimulated. There are many TLRs that we have in our immune host system, 1 through 9. And this is actually stimulating TLR 7 and 3 to see if they have synergistic or adverse effects. And you can see that when we combine certain TLRs, it can have a beneficial synergistic response that will broaden our cytokine response. Next, please. So the most clinically relevant that we see have been TLR4 and TLR9. And these adjuvants that we have studied are MPLA and CpG. When in combination and individually, we have studied if they are beneficial and give us that synergistic immune response via interferon beta and IL-12p70 that we have been looking on. Thank you. The diagram that you see is comparing different densities of the adjuvants onto the particle and see if they give us various response. In our diagram, we see that the higher density of adjuvants actually produce a higher immune response that we are looking for. Next, please. So in this regard, we started to look at the various aspects of the toll-like receptor pathway and how they can talk with each other. Next, please. Here are ELISAs we did out of knockout mice to go down from the top to the bottom of how we see our signaling pathway. So if you look on the left-hand side, we took MIghty88 knockout and TRIF knockout. So MIghty88 would be TLR9 and TRIF knockout would be more TLR4, and see if affecting the very top would affect the downstream cytokine profile. If you see at the left, MIghty88 has a greater impact than our TRF knockout. Also with our IL-12p70, we had the same response, whereas MyD88 globally knocked out all our cytokine response. Our TRIF knockout still had some response, meaning they still were not completely ablated and had various other crosstalk methods. Next, please. Going down the pathway, we looked at a downstream transcriptional factor called interferon factors. And these are interferon 3, 5, and 7. Canonically, in our literature right now, interferon 3 and 7 seem to be the main immune responses. However, currently, or in the last couple years, our main focus has become interferon 5. As you see in the middle diagram with the IRF5 knockout, with any stimulant that we put to it, whether it's TLR4 or 9, it was globally ablated. Next slide, please. And this is also seen with the IL-12P70 as well. Next, please. So from all the diagrams that I've showed you, we have come to our main conclusions that, one, our MyD88 adapter protein is very critical in the signaling for CpG-induced TLR signaling. Our TRIF pathway and signal is required for any synergistic boost or signaling between our TLR4 and TLR9 pathways via MPLA or CpG. And looking at the immune response via IRF3, 5, and 7, our IRF5 seem to be our most critical transcription factor for synthesizing and synergistic innate immune response via MPLA and CpG. So this, we have explored more of the IRF5 in our downstream pathway. Next, please. So here we are looking more at the biochemical properties via protein analysis and ELISA of our signaling pathways. Next, please. So if you look on the left-hand side, this gives us our complete pathway. So I know I threw a lot of acronyms and signaling pathways at you. So if you look on the left-hand side, you'll have our TLR4 pathway, which will go through MyD88 and subsequently TRIF6, TAC1, and to IRF5. Also, we believe that endosomally, the TLR4 and TLR9 has that synergy that you see on the right-hand side, and that's what we are seeing with our synergistic increased immune response. And that is via TRIF and MyD88 on the right-hand side. Thus, we need both to have the increased immune response that we require to host long protective host immunity. Next, please. Here, we have been exploring more of the IRF5 activation and the kinetics in a fashion of stimulation with 30 minutes, 1 hour, 2 hour, 4, 6, and 24. The Western blot that you see currently in between on the left is a newer technology that has been put out in the last couple of years called a FOSTAG technology. This technology is used for antibodies that do not readily have a commercial antibody, such as IRF5. This will also show us the multiple phosphorylation sites that can occur with activation of that protein. On the left, the first part, you will see the single adjuvant of MPLA, which will be TLR4. So you'll see that signaling at half an hour and 1 hour, which ablates more after 2 hours. Underneath, you'll see CpG, which will be TLR9. And most interesting, below, at the bottom, you'll see a combinatorial TLR4, TLR9 signaling, which increases the phosphorylation event from 30 minutes to 1 hour, all the way up to at least 4 hours. And this is indicative of how we have that broadening immune response from that IRF5 protein. If you look on the right, that's from the paper quoted and how this signaling event is supposed to be seen for the phosphorylation. Next, please. Here, we wanted to see more of our activation and nuclear translocation of IRF5. So IRF5 in the cytosol will be activated, phosphorylated, and then nuclear translocated, and then form a dimer in the back up into the cytosol. So when we did nuclear fractionation of mouse dendritic cells in this kinetic fashion, we saw that at 4 hours, we saw an increasing amount of transcript in the nucleus in the dual TLR4 and TLR9 pathways compared to the single pathway, more adding credence to our synergistic response between TLR4 and TLR9. Next, please. And here for our, after our nuclear fractionation, we did back to our cytokine panel to see where they peaked. And here we can see that in a synergistic fashion, we can see interferon beta peak at 6 hours and IL-12P70 peak at 12. So this correlates to the concept of you have phosphorylation early on, you'll have nuclear translocation at 4 hours, and then you'll have the production of the cytokines of interferon beta and IL-12P70 subsequently afterwards. So this helps us explore a model to help make our vaccines more targeted and more efficacious, to have a broad synergistic effect and have less side effects in the future. We have found through our studies that MIT88, TRIF, and IRF5 are all essential for innate immune responses and synergy between MPLA, which is TLR4, and CpG, which is TLR9. These combination adjuvants can result in a sustained gene expression that we see via our protein expression and our ELISA. Our future work will consider going and identifying other key signaling proteins in the signaling pathway, such as TRIF and TRAF6. And this can be also used in other bacterial and viral pathogens that we see in current life. Next, please. I'd like to acknowledge all my co-authors and collaborators from Georgia Tech, Randy, Palab, Ameline, Neha, Gabriella, Cole, Dr. Santangelo, Dr. Shakhmatov, and Dr. Roy. I'd like to especially thank my mentor, Dr. Shakhmatov, for his support in teaching, and the Lowen Center, Emory, and Georgia Tech. Thank you very much.
Thank you so much, Dr. Gita. That was a very sophisticated project. Really great work. I'd like to open up the questioning. Please feel free to enter any questions in the chat feature. But first, I'll turn to our judges. Any questions from the judging panel that you may speak if you choose?
Hi, this is Anne. That was a great presentation. I had a question about sort of your future directions. And I was wondering if you have any plans to test either these adjuvants, either alone in combination with the particular vaccine of interest. So whether you can see if they actually enhance the specific immune responses to either the bacteria or the virus, you know, whatever it would be combined with in a vaccine.
Yes, so our group has done a lot of influenza studies in the vaccine model, and I want to currently take this IRF5 model that I am currently studying and implicate it into other diseases which currently are in press, such as autoimmune, cancer, any inflammatory diseases, and just macrophage polarization. So I want to explore the aspect of IRF5 being that central regulator, just like NF-kappa B that we've had in the past.
Thanks.
Okay, thank you.
Any other questions?
This is Rabin. May I ask a question to Navdeep?
Yes, please, Rabin.
Hello, Navdeep. Thank you for sharing your data. You did a very impressive set of studies. I have a question for you regarding the representativity of mouse models in vaccine research and sort of immunology in general.
Yes.
I mean, as you're probably familiar with now, there is this recognition that wild mice are perhaps much closer to humans than lab mice, and there are some attempts to sort of mimic that by the idea of collaborative crosses in particular to sort of build some diversity back into the animals. But I'm more interested in sort of what you think is the immune poise of lab animals versus wild animals with regards to some of these pattern recognition receptors. So how do you think this is tuned in vivo when we're sort of exposed probably to many more pathogens all the time and PAMPs, as opposed to the jailed but sort of cozy life of a lab mouse?
That's a very good question, considering most everyday person is indoctrinated with a lot of pathogens all the time, so we are constantly inflamed. So yes, it is a little skewed in how you see a cleaner mouse, as you said, in the lab and the responses, but we need to start at some base point. So once we can identify the inflammatory markers in these base mice, then we can explore into a in a more quote unquote dirtier, cleaner mouse to have everyday pathogens to see if we'll have the same effect. But it would definitely be a point of interest in the future.
Thank you. And with that, we'll thank Dr. Gita again, and we'll transition to our next speaker.
Thank you.
Clint?
Yes, next we'll hear from Dr. Veronica Obregon-Perco, who is a postdoctoral fellow in the Division of Infectious Diseases. She's been here at Emory for 2 years and received her PhD from the University of Texas Health Sciences Center in San Antonio. Her mentor is Dr. Ann Cerruti, and her research focuses in viral recrudescence in pediatric HIV infection. Veronica?
Hello, can everybody hear me okay?
Yes.
Okay, great. Well, I'd like to first thank the organizers for allowing me to share my screen instead of using the slide deck. I decided to be a little extra and have animations, and so this is just going to be the easier way to go about it. So thank you to Carol and Barbara for helping me with that. So today I'll be talking about a study in our lab aimed at uncovering the hiding places of the HIV-1 reservoir using a pediatric non-human primate model. And so mother-to-child transmission continues to contribute to new infant infections of HIV-1 worldwide, as there were 150,000 new cases last year alone, now adding to the nearly 2 million children living with the disease globally. And current art-based strategies to prevent mother-to-child transmission are limited by implementation challenges, including late presentation to prenatal care and poor adherence. And so this contributes to maternal infections going untreated in the peripartum period, creating a risk for transmission to the infant through breastfeeding. And in fact, it is now this route of transmission that accounts for up to 75% of new infections worldwide. And so the major obstacle to HIV cure is the early integration of HIV provirus into infected cells. And these cells are not cleared by antiretroviral therapy. And so their persistence contributes to a long-lived reservoir, which is capable of reactivation following ART cessation. And so for a long time, HIV researchers have been playing hide and seek with the virus and trying to uncover the cell types and the tissue sites of where the reservoir hides so it can be targeted by cure strategies. And much of what we've learned in this area has been gained from studies in adults or in animal models of adult age. And we know that pediatric immunity and HIV-1 infection is distinct from that of adults, including in routes of transmission and tempo of disease, all of which could potentially impact how the reservoir is established and how the reservoir hides differently than what we would learn from in adults. And so in order to make strides in pediatric cure efforts, it's important that we get an accurate picture of the reservoir in the pediatric setting. And really, we can make terrific movements in this area with the use of animal models, which will allow us to access sites that would be otherwise difficult in a clinical study. And as you heard in our keynote today, the model of choice in the Cerruti lab is the simian immunodeficiency virus, or SIV, infection of rhesus macaques, because it models so many aspects of HIV pathogenesis and reservoir establishment. And in fact, we've previously used an infant macaque model of oral SIV infection and long-term ART to evaluate sites of persistence. And more recently, we're also using this model to evaluate cure strategies. And so we sought to confirm and expand this model and use it to assess reservoir composition, this time using a chimeric virus referred to as a SHIV, or simian human immunodeficiency virus, which is an SIV that can replicate in macaques but expresses the human HIV envelope, and you can allow you to do direct testing of HIV envelope targeting strategies. So in our study, we had 4-week-old macaques that were orally infected with this SHIV in 2 doses. And after 8 weeks of infection, they were placed on a year-long ART regimen. And at that point, the animals were separated into 2 groups. One group was sacrificed on therapy for reservoir characterization, and the other group proceeded to treatment interruption to assess viral rebound, but I'll primarily be talking about the first group today. So all of our infant macaques were successfully infected, and you can see those here in blue. And so you can see that there's a peak viremia, but then this kind of slow decline to set point and this sustained viremia, which is actually a feature of what's seen in HIV-infected children as well. Meanwhile, there on the right in red, you can see adult macaques infected with the same SHIV, and they exhibit more of what we see in adult HIV infection, and that is the typical peak and rapid decline to set point. So really capturing what we see in the clinical setting in our infant macaques. And so for the subset of animals that were sacrificed on therapy, you can see once they started their antiretroviral therapy, the viral loads became suppressed and remained below the limit of detection for months prior to the endpoint of our study. And most of the animals didn't show any disturbance of the CD4 T cell pool in terms of frequency, but there were 2 animals that did show some decline in CD4 T cell percentages, but those typically restored after ART administration. So first I'll show you some data on anatomical sites of viral persistence. So we longitudinally tracked viral DNA in CD4 T cells that were isolated from the blood, lymph node, and rectal biopsies. And you can see in the blood and lymph node, there is a decrease in viral DNA after ART administration. And in rectal biopsies, we see something of a stability from time of early ART initiation to later on in the regimen. And post-mortem, we also had the opportunity to assess levels in the brain using myeloid cells. And you can see compared to the other compartments, the levels were largely undetectable. So we asked if this viral DNA represented a replication-competent virus rather than a large accumulation of defective virus. And we did this doing a viral outgrowth assay, which measures virus capable of infecting uninfected cells. And here you can see that we are able to recover replication-competent virus from the blood as well as the spleen to represent lymphoid organs. We didn't have enough cells to do all compartments, but I think this demonstrates that the DNA we're detecting does reflect levels of replication-competent virus being present as well. In addition, we also saw that the 2 animals that were below the limit of detection in lymphoid compartment were also two of the animals that had some of the most dampened viral loads. So suggesting this burden in this tissue site might be more reflective of plasma events prior to ART initiation. We also quantified RNA levels longitudinally, which would be an indicator of a more active reservoir producing viral RNA products. And so you can see again that there is a decline in blood and lymph node CD4 T cells after ART initiation. But we also noticed that after a year on the regimen, that there seemed to be a higher level of viral RNA in rectal biopsy CD4 T cells compared to lymph node and blood. And furthermore, there was also stability of this RNA from early in the regimen to late in the regimen. Whereas when we look at another compartment like the blood, we can see that even during the regimen, there's a continued decay as where it's more stable in the rectal biopsy compartment. Now, in the previous slide, I showed you that this isn't because of more infected cells in this compartment per se, because there were similar levels of viral DNA at the same time point. And in fact, when we calculate RNA to DNA ratios, we see that that ratio is significantly higher in the rectal biopsy compartment. So per DNA molecule, there is more RNA transcription, suggesting that this site is an area of active virus transcription under ART suppression relative to the other sites. And so those are anatomical sites of persistence, but what about cellular sites of persistence? So we've become interested in evaluating the role of various CD4 T cell subsets in viral persistence in pediatric infection. So when we turn to the literature, we see that virus can be harbored in a variety of resting memory T cell subsets, but generally central memory and effector memory CD4 T cells have been regarded as important reservoirs because they're enriched for HIV or SIV DNA in macaque models. And we see now with emerging evidence that levels of HIV-1 provirus, not necessarily total DNA, are pretty similar across CD4 T cell subsets, and that the levels of reactivation of this provirus are also similar across memory subsets. So challenging the notion that central memory and effector memory may not be the only key players. In addition, there's new evidence to suggest that naive CD4 T cells, which have generally been disregarded as a reservoir because they're not a memory cell, can also harbor virus capable of reactivation. And this cell type is of particular relevance in the pediatric setting because it dominates the CD4 T cell pool in infancy. So we sought to address these findings in our model. So we sorted various T cell subsets from CD4 T cells in the blood and lymph node compartments and quantified the level of viral DNA within each subset. And consistent with the literature, we do see an enrichment of viral DNA in effector memory cells, as well as follicular helper cells of the lymph node. However, we also see DNA in naive CD4 T cells at comparable levels to central memory CD4 T cells in both compartments. So when one considers the frequency of infection, as well as the frequency of that particular subset in the CD4 T cell pool, we can calculate their contribution to total DNA persistence. And when doing that calculation, we can see that naive CD4 T cells, shown in blue, actually make up the majority of the total SHIV DNA persistence in blood and lymph nodes. But as I mentioned before, in chronic HIV infection, the DNA landscape can be comprised of a large majority of defective viral DNA or defective provirus. And in fact, we're learning that it's actually only a small fraction of viral DNA typically represents intact provirus capable or presumably capable of reactivation and reseeding the reservoir. And an even smaller fraction actually does become reactivated in a culture setting. So we sought to assess the reservoir composition within naive CD4 T cells and bulk memory CD4 T cells by quantifying the levels of intact genomes in each of these cell types. And we can see that, consistent with the DNA findings, there is an enrichment of intact genomes within memory CD4 T cells. However, there are still intact genomes within the naive CD4 T cell compartment as well. And again, this cell type is also more abundant than memory cells. In addition, when we calculate the ratio of intact provirus to total DNA, we see an average 1:2 ratio, meaning for every 2 total DNA molecules, one of those represents an intact provirus. And we weren't able to do the same calculation in our bulk memory cells because we didn't have the total DNA to do the calculation. However, a recent report approximates that about 60% of total viral DNA is intact provirus in CD4 T cells from the lymph node of adult macaques, which is going to be an age where memory cells will be more abundant. So in a way, this calculation gives us an opportunity to gauge what that percentage could look like in our memory CD4 T cells. And it would suggest that the fraction of intact provirus within naive CD4 T cells is likely comparable to memory cells based on this report. So in summary, I've shown you that after a year of ART, SHIV DNA is detectable at comparable levels across blood, lymphoid, and rectal CD4 T cells with not much in the brain, and that blood and lymphoid organs harbor replication-competent virus, but the levels in the latter compartment seem to be more reflective of pre-ART plasma viral burden, suggesting it's an early site of infection and possibly a source of plasma viremia in the absence of ART. We also show that the rectal compartment is a site of active viral transcription under ART-mediated viral suppression, which could suggest that it's a tissue sanctuary that allows this ongoing low-level transcription, and that it could possibly be an early site of viral reactivation following ART interruption. And lastly, coupled with their frequency of infection and their abundance in the T cell pool, naive CD4 T cells make a substantial contribution to total SHIV reservoir burden in ART-treated infant macaques. And so they will definitely be something that needs to be further studied, but may also need to be considered in pediatric cure efforts. And so ongoing studies in our lab are aimed at expanding our analysis of active viral transcription to other sites of the gastrointestinal tract outside of just the rectum, and also determining the anatomic origins of viral rebound following ART interruption and asking the question if the gut plays a role in that reseeding of the reservoir. And lastly, to evaluate the contribution of CD4 T cell subsets not only to persistence, but also to viral rebound as well. And so I'd like to acknowledge the rest of the members of the Sharuti Laboratory, our collaborators, and our funding sources, and I'll be happy to take any questions.
Okay, thank you so much. I think we have time for one question. Would one of the judges like to ask a question? Dr. Carpin, would you like to ask one?
Sure, I'll do my best, but it's totally as an amateur. But I have this really nice presentation. I'm sure you're aware that Cassie, one of your colleagues, is doing a study of this in men, you know, on sort of rectal transmission. And I wonder what sort of the correlates you might have for kind of human issues about this higher persistence, it looks like, in the rectal mucosa.
Right, yeah, so I have seen some literature to suggest that usually in HIV infection, there seems to be, the gut seems to have been identified as actually a lower area of RNA transcription. And so it's unclear whether that's because of duration of ART regimen or type of ART regimen, or in our case, whether it's because of age or type of challenge. So it's definitely still not entirely clear if that's a universal finding or if it's going to be unique to the pediatric setting or unique to the oral transmission route as well. We are getting some preliminary data to suggest that upon ART interruption, the gut is one of the first areas to reactivate and contribute to plasma viremia. So that's another aspect that we're pursuing as well.
Thank you.
Thank you so
Thank you.
much. I really appreciate you explaining that again, another sophisticated project, very well explained. So with that, we're going to take a short break. We plan to resume the program at 2:15. So go take a quick bio break, and we'll be excited to come back to our basic science poster blitz. So please don't be late. It'll be lots of fun. We'll see.
Well, it's time to get started again, and this portion of the program will feature abstracts selected for basic science poster presentations. We'll use the same format that we did in our earlier poster blitz, but for those of you who are new to it or new to the meeting, we'll have 6 presenters who have recorded short 3-minute talks highlighting the key points of their abstracts. And these will be presented in rapid succession without any question and answer. But then between 3:15 and 4, we'll have a poster Zoom session where you will be able to go to the individual speakers' Zoom room and have a conversation with them. And those Zoom rooms can be found on the program listing as well as in the abstract booklet. So we'll begin these with our first basic science presenter, who is Dr. Holly Bowser-Heaton. She is currently a K-12 scholar and an interventional cardiologist on the faculty of in cardiology. And the title of her talk is Patient-Specific 3D Bioprinted Platform for In Vitro Disease Modeling and Treatment Planning in Pulmonary Vein Stenosis. And I don't think we're hearing the sound.
Thank you for the opportunity to share our work for 3D bioprinted platform for in vitro analysis of pulmonary vein stenosis. This is a collaborative effort, as you see here. Pulmonary vein stenosis is a cardiovascular condition primarily affecting children, characterized by progressive lumen size reduction of one or more pulmonary veins, which leads to unfavorable patient prognosis at advanced stages, and therapies are limited. Animal models that rely on tying off a pulmonary vein may not replicate its causes and are less tractable with respect to genetic and microenvironmental manipulations, complicating translation to clinical therapies. And a 3D in vitro model of human pulmonary vein stenosis and pulmonary vascular disease in general that is capable of vascular perfusion currently do not exist. Therefore, in this study, we aim to create a relevant, patient-inspired model for the study of flow and geometry and investigate the cellular changes that occur in response to these modifiable factors. Our experimental methods are as follows. We used a clinical model based on CT or X-ray angiography and generated a 3D model, an STL file, of the pulmonary veins. Gelatin methacrylate at 20% with added LAP was prepared and used for the DLP bioprinter to make a bioprinted model. The then assembled hybrid pulmonary vascular stenosis bioreactors were then tested under flow conditions that are clinically relevant. For the healthy model, we used 150 mLs per minute and the stenotic model 50 mLs per minute to replicate clinically seen flows. Cytoplasmic GFP labeled human umbilical vein endothelial cells were then used to line the models and were tested under static and flow conditions. And finally, computational fluid dynamics was used for modeling and analysis, with particular attention to wall shear stress, velocity, and pressure in all of the models with various geometries as mentioned. Here are results. On the left, you see the healthy model with no stenosis with a replicated bifurcation lesion. On the left, you see DAPI-tagged endothelial cells do in fact line the entire bifurcation of vascular tree, and Connexin 43 shows that these cells do form gap junctions. In the center, you see the stenotic lesion. And I want to particularly pay attention to region 4, and this is the area of decreased lumen size simulating pulmonary vascular stenosis. You see here that the endothelial cells do in fact replicate at a higher rate, and therefore the lumen size decreases further. This is what we see clinically as well, and we're excited about this result. Finally, on the right, you see the computational fluid dynamic analysis of our models. As you can see, velocity and wall shear stress are all increased at the area of stenosis. And pressure is also increased upstream of the stenosis as seen clinically. Thank you so much.
All right, our next presenter is Dr. Alexander Camarada Mushtours, and he is a postdoctoral fellow in the PACS division studying cystic fibrosis. And the title of his talk is Epigenetic and Transcriptional Dynamics in Acute and Chronic Human Myeloid Inflammation.
Hi everyone, my name is Alexander. I'm a postdoc at the Tiruvanziam lab. I'm coming from a fundamental research background studying the dynamics of NF-kappa B pathways in Drosophila, and I'm now transitioning into translational research. The background of this project begins with a study on cystic fibrosis. The Tiruvanziam lab was able to identify a new type of neutrophil in the lungs of CF patients. Those neutrophils are reprogrammed transcriptionally, and they are enabled to clear the bacteria present in the lungs of CF patients. Using a narrow air liquid interface model, we're able to reproduce this phenotype and obtain the reprogramming. Using the same in vitro assay, we're able to obtain some inflamed lung recruited infected neutrophils in the context of SARS-CoV-2, which might prove useful to understand the cytokine storm that has been described. In the juvenile arthritis disease, we also observed a difference between the neutrophils that are in the blood compared to the ones that are present in the synovial fluid of patients. The ones that are present in the SF indeed showed some transcriptional reprogramming, and they are the ones responsible for the damage to the joints. So 3 different pathologies, but one common point: the transcriptional reprogramming of neutrophils that are recruited at the pathogenic sites, and that leads to the establishment of an inflammatory process in an acute or chronic manner. In our transcriptomics data, we were able to identify several epigenetic related genes. Among them, some histone deacetylases or HDAC, and I'm showing you here 10 different HDAC that are differentially expressed over time in CF contexts in neutrophils. We focused on one of them for a pilot experiment, HDAC11. HDAC11 because it has already been proved useful as a treatment for cancer. We used a known inhibitor, Cis17, and we're able to restore the antibacterial response of the GRIM neutrophils in CF, thus restoring their phenotype. In a more perspective point of view, we are showing that in different contexts, neutrophils are more plastic than what previously described. Neutrophils are able indeed to dynamically regulate their response through the action of histone deacetylases. Those HDAC have an expression that is controlled in a time manner in the cell. Using specific compounds, we could target specifically the temporality of the expression of HDAC, thus affecting the temporality of the cell response, thus modulating the host response to these pathogens. Using an array of assays at different omics level, would then be able to assess all of that, and we'll use computational integration to get a multi-omics vision of the response. And now I'm ready to take your questions.
So you can visit Alexander's Zoom room and ask him those questions. Next, we'll hear from Dr. Satish Chonat. Satish is a current K-12 scholar and is a pediatric hematologist in AFLAC. The title of his study is Complement-Mediated Acute Lung Injury in Sickle Cell Disease, Novel Mechanisms and Therapies.
Hello, I'm going to brief you on my abstract titled Complement-Mediated Acute Lung Injury in Sickle Cell Disease, Novel Mechanisms and Therapies. Acute chest syndrome is a frequent complication in children with sickle cell disease. It has the highest incidence in children, remains the leading cause of admission to the ICU, and accounts as many as 25% of deaths in sickle cell disease. We noticed that in these patients, markers of alternative complement pathway were significantly elevated during acute chest syndrome in comparison to their baseline. Steady state is shown in this figure. On the right is a complement cascade where the highlighted complement proteins toxins were the ones which were analyzed in the study. Our objective was to characterize key molecular factors that play a role in its pathogenesis in order to identify targets to prevent or treat ACS. So to study this further, we used two models. One is the human-induced acute lung injury and a novel cobra venom factor. Cobra venom factor has been used in immunology to activate and then deplete complement. It's structurally similar to complement protein C3, which can create an amplification loop, thereby activating the terminal pathway and subsequently formation of membrane attack complex. And we injected these mice with various agents, and then we used this MOSOC, which is a device to assess acute lung injury. We attach a small collar to the neck of these mice, and they can detect heart rate, respiratory rate, or breath rate, oxygen saturation. After injection of one of these agents, these mice are monitored for up to 2 hours or till they die. At the end of the study, we collect blood, bronchoalveolar lavage fluid, and harvest lung for imaging. Complement activation in these mice seemed to result in marked hemolysis, increase in the plasma and BAL aflatoxin C5a, and also we saw the complement deposition in the lungs. Sickle mice develop frequent desaturations and eventual death anywhere from 10 minutes to 2 hours after CBF injection. Each black or blue bar denotes a mouse, and as shown here, the oxygen saturation decreases to less than 50% before dying. Conversely, control mice do not exhibit significant ALI. More importantly, these complement activations are universally fatal to sickle mice and not to their controls. On the right, human treatment of sickle mice results in ALI and rapid death, as shown in the red dotted line. While our novel global complement protein C3 knockout of sickle mice appear to be protective from human-induced ALI. More to come. Thank you.
Our next presenter is Jamie Story, who is a PhD candidate in molecular and systems pharmacology, working in the laboratory of Dr. Sunil Rekar in AFLAC. The title of her study is bortezomib significantly enhances gamma delta T cell mediated lysis of acute myeloid leukemia and T cell acute lymphoblastic leukemia.
Hi everyone, my name is Jamie Story and I'll be presenting data from the Rekar lab on how bortezomib enhances gamma delta T cell cytotoxicity of AML and TLL cells. As most of you know, relapse is still a clinical barrier for childhood leukemia, highlighting a need for the development of more therapies. Our laboratory is interested in utilizing gamma delta T cells as an immunotherapy for AML and TLL due to their natural mechanisms of inducing cancer cell lysis through engagement with stress antigens. Certain drugs, such as the proteasome inhibitor bortezomib, are known to increase surface expression of these stress antigens on cancer cells. Bortezomib has proven to be safe in humans in AML clinical trials, but ultimately was ineffective as a single-agent therapy. We hypothesize combining bortezomib with gamma delta T cells will be an effective immunotherapeutic approach for AML and TLL. To explore this hypothesis, we first determined the optimal dose to elicit increased surface expression of stress antigens on AML cells. We then treated the AML cells with this dose or vehicle control for 24 hours before incubation with gamma delta T cells and a 4-hour flow cytometry-based cytotoxicity assay. Next, to see whether bortezomib treatment accelerated target cell death, AML cells were co-incubated with gamma delta T cells for 48 hours with the assessment of percentage of remaining live cells at 4, 12, 24, and 48 hours. We repeated these experiments with two T-ALL cell lines to investigate whether results were repeatable in the T-ALL setting. We found that bortezomib treatment significantly increased the stress antigen ULBP256 in both AML cell lines, Kasumi1 and NOMA1, and that the bortezomib treatment significantly increased target cell death, particularly at lower effector to target ratios, which are more clinically relevant. We also found that bortezomib treatment significantly decreased the remaining percentage of live NOMA1 cells at 24 and 48 hours at a 1:2 E/T ratio when compared to vehicle control treated NOMA1 cells. When we repeated these experiments into TAL cell lines, GERCAT and MOLT4, we also saw a significant increase in ULBP256 with bortezomib treatment and a significantly higher cytotoxicity of target cells at the lowest 2E/T ratios. We also saw that the remaining live GERCAT cells in a 24-hour cytotoxicity assay were significantly lower with bortezomib treatment at a 1:4 and a 1:20 E/T ratio. Overall, our results show the combination of bortezomib and gamma delta T cells is a promising immunotherapeutic approach for AML and CLL. Thank you. Hi, everyone. My name is Jamie Story.
Okay. Our next presenter is Dr. Jessica Raper. Jessica is an assistant research professor at Yerkes in developmental neurosciences, and the title of her study is Post-Exposure Prophylaxis to Mitigate the Neurodevelopmental Consequences of Postnatal Zika Virus Infection in Infant Rhesus Macaques.
I'm Jessica Raper. I'm going to talk about our project with Dr. Trudy looking at post-exposure treatment to mitigate the neurodevelopmental consequences of postnatal Zika infection in infant monkeys. So Zika infection in adults is typically mild, but during the 2015 outbreak, it was shown that Zika can disseminate into the brain, causing damage, and particularly during in utero infection. However, very little is known about how it may impact the rapidly developing postnatal brain. So using a rhesus monkey model, we have shown that Zika infection during infancy negatively impacts brain development, resulting in long-term behavioral, cognitive, and motor impairments. And a recent rodent model of in utero infection shows that sofosbuvir treatment given to the dam prevents viral dissemination to the fetus. So the purpose of our study was to look at whether this treatment given after exposure could mitigate the negative impact of Zika virus during infancy. Using 3 infant rhesus monkeys infected with Zika at 5 weeks of age, followed by a 14-day post-exposure treatment with sofosbuvir. We have peak viremia at 2 to 3 days, with viral clearance at 7 and 10 days post-infection. We then followed the animals for 12 months, looking at growth, social behavior, emotional reactivity, and cognitive function. First, looking at growth, we have the Zika plus sofosbuvir treated animals are the triangles with a purple line, and we can see that they have comparable head circumference and body length development over the 12 months of age compared to nursery-reared and mother-reared controls. Next, looking at social behavior, we see them interacting in a large novel social cage, and we see that we looked at their latency to enter this cage. While Zika-infected infants, shown in a dashed line, took longer to habituate to this novel environment, those with sofosbuvir treatment looked more similar to the controls. Emotional reactivity was a little different story. Using the human intruder paradigm, we see species-typical increased freezing during this profile condition from the control animals, whereas the Zika-infected infants did not exhibit this behavior, and the sofosbuvir treatment animals have an intermittent phenotype showing some freezing, but not comparable to that of controls. Lastly, looking at cognitive functioning, we see a delay-dependent memory impairment in both the Zika and those treated with sofosbuvir. However, sofosbuvir did spare visuospatial processing such that they had a comparable level of performance on this task here as compared to the controls. So overall, we have an intermittent phenotype in these animals, and thank you for listening.
Okay, our final poster presenter is Dr. Patricia Zera. Patty's a current K-12 scholar also, as well as a pathologist and hematologist. She will present her study entitled Factor VIII Specific CD4 T cell proliferation requires multiple previous exposures to Factor VIII.
Good afternoon, and thank you so much to the organizers for letting me talk to you about my poster presentation showing that factor VIII specific CD4 T cell proliferation requires multiple previous exposures to factor VIII. Factor VIII replacement in hemophilia A can be complicated by neutralizing IgG alloantibodies known as inhibitors that block factor VIII activity and prevent optimal replacement. And in contrast to other model antigens, inhibitor formation occurs only following multiple factor VIII exposures, both in human and mouse models, suggesting that early exposure events may prime the subsequent development of long-lasting antibodies. And while little is known about the immune events underlying inhibitor development, previous studies suggest that CD4 T cells play an important role in inhibitor development. However, the timing and overall role of these CD4 T cells in the immune response to factor VIII remains incompletely understood. Therefore, we have two objectives in our study. The first was to develop a unique immunologic tool for examining the factor VIII specific CD4 T cell response following factor VIII exposure. And the second was to then use this tool to examine factor VIII specific CD4 T cell activation and proliferation following factor VIII exposure using a hemophilia A mouse model. First, we engineered a portion of the common model antigen OVA into the B domain site of B domain deleted factor VIII to create a novel factor VIII replacement product termed HOVA. To utilize HOVA, splenocytes are isolated from a CD4 T cell receptor transgenic OT2 mouse with the T cell receptor specific for the OVA peptide, then labeled with CFSE, allowing us to detect their proliferation, and then adoptively transferred into a hemophilia A mouse model, which is challenged with HOVA administration. Then we're able to examine splenocytes at various time points after HOVA is administered, allowing evaluation of factor VIII specific CD4 T cells. We first evaluated OT2 T cell division after hemophilia A mice were exposed to either 1 or 2 injections of HOVA following OT2 adoptive transfer. And we were surprised to see that there was no evidence of proliferation or activation on day 5 following exposure to the antigen. This is unlike other common antigens we have evaluated using this system, where proliferation is usually observed after just 1 exposure to the antigen. We subsequently performed adoptive transfer of OT2 cells into mice that were previously highly immunized to factor VIII and saw that there was significant proliferation and evidence of activation marker upregulation after just 1 exposure to HOVA. We speculate that the presence of perhaps immune complexes in highly immunized mice may help to accelerate the immune response against factor VIII. However, future studies are clearly needed to elucidate the exact immune mechanisms underlying this phenomenon. Thank you so much for your attention, and I'm looking forward to talking to you all in the live poster presentation following the recording.
Yeah, so speaking of that, we will go back to the poster Zoom rooms at 3:15. And in the meantime, we have 2 more oral presentations. The first is by Dr. Catherine Oliver, who is a postdoctoral fellow in the PACS division. Catherine has been with Emory for 4 years. She received her PhD from the University of Alabama, Birmingham, before coming here to work with Dr. Eric Sorcher on cystic fibrosis research. So the title of her talk is The Discovery of Specific Ribosomal Proteins as Investigational Therapeutic Targets for Cystic Fibrosis. Catherine? You need to unmute. Barbara, can you help her?
Try it. Okay. Can you hear me now?
Yes.
Okay. Yeah, I, for some reason, didn't have the control to select unmute, but it's working now. Thank you for the kind introduction and for the opportunity to present this story to you today. It's going to describe a bit of a unique path that our laboratory has embarked on in the pursuit of more effective therapeutic options for patients with cystic fibrosis. It includes a number of model systems such as yeast, primary human airway epithelia, and mouse models, and arrives at where we are today with preclinical evaluation of an investigational anti-ribosomal protein intervention. There's only one disclosure that's relevant to this presentation, which is that I am currently engaged in a scientific collaboration with Ionis Pharmaceuticals, but otherwise nothing else to disclose. And Barbara, okay, I think it's working, yes. A brief introduction to cystic fibrosis. It is a lethal autosomal recessive disorder that affects approximately 70,000 people worldwide. Multiple organs are involved in the disease, with the most common clinical manifestations including digestive malabsorption, pancreatic dysfunction, and chronic pulmonary infections, among others. CF is caused by loss of function of a single gene called CFTR, or the CF transmembrane conductance regulator. It's a large ABC transporter encompassing nearly 1,500 amino acids with 2 large transmembrane domains, or TMDs, 2 nucleotide binding domains, or NBDs, and 1 R domain whose structure has not been resolved to date. So it's represented by a simple pink ribbon structure here. CFTR functions as a chloride and bicarbonate channel expressed at the apical surface of secretory epithelia, thereby facilitating transepithelial transport of these ions. Unfortunately, more than 1,600 disease-associated variants have been reported to date, which renders personalized medicine approaches to the treatment of CF quite difficult. In an attempt to address this issue, investigators have traditionally binned CFTR variants into 6 different subclasses based on molecular phenotype. Under normal conditions, CFTR is synthesized in the nucleus, chloroglycosylated in the ER, fully glycosylated through the Golgi, then trafficked to the cell surface. Mutations have been identified at each of these steps, so I've just listed a few here that will be discussed further in the presentation. The most common CFTR variant is F508del, or delta F for short, which is a class 2 mutation that exhibits a folding defect and is recognized by ER-associated degradation. Thus, the polypeptide is trafficked to the proteasome, resulting in little to no functional protein reaching the cell surface. Another pertinent mutation class that encompasses roughly 10% of the CF population includes class 1 variants, which are largely represented by premature termination defects. Examples include G542X and W1282X CFTR. These variants are primarily subjected to mRNA degradation via nonsense-mediated decay, which results in significantly decreased mRNA levels and little to no protein. Recent advancements in personalized medicine have led to FDA approval of some CFTR modulators, with the first example here being Kalydeco, or VX770. It's a potentiator that is designed to bind cell surface resident CFTR and promote the opening or gating activities of the channel. The next cohort include Orkambi, Symdeko, and Trikafta, which are combination therapies that implement both this VX770 potentiator as well as a CFTR corrector, such as VX809 or VX661 that are listed there. These small molecules are designed to bind cytoplasmic CFTR and promote a more native-like folding trajectory such that more CFTR protein reaches the cell surface, where the potentiator can then bind and further amplify the CFTR gating. CF patients approved for Kalydeco, Orkambi, Symdeko, and/or Trikafta have experienced some improvement in clinical endpoints, such as increased lung function, but there is still a need for more robust therapeutic interventions. Moreover, patients who, we went too far, patients with premature termination alleles remain without effective treatment strategies. And thus, our laboratory decided to pursue a novel approach to these issues by utilizing yeast phenomic analyses to identify genetic modifiers of mutant CFTR biogenesis. This was done in collaboration with the Hartman Laboratory at the University of Alabama at Birmingham. So yeast express a homolog to CFTR, also an ABC transporter, termed YOR1, which means yeast oligomycin resistance 1. It normally functions as an oligomycin efflux pump, and as you can see, possesses a tertiary structure that's very similar to CFTR. In addition, mutations analogous to the F508del, G542X, and W1282X variants in CFTR also occur in UR1. These are annotated as F670del, G704X, and L1282X, respectively. These 3 UR1 mutations were individually screened, meaning that each one was crossed with 5,000 single gene deletions from the yeast deletion strain library, and the resulting haploid double mutants were assessed for enhanced resistance to oligomycin. So in other words, we were interested in those gene deletions that could rescue or overcome the F670del folding defect and or the G704X or L1282X premature termination defects. These are examples of the F670del haploid double mutants evaluated, where the colonies were plated on normal yeast growth media called YPEG, and media, the YPEG supplemented with oligomycin. Examples of colonies that survived in the presence of oligomycin are indicated in red. We identified several gene deletions that would be expected, such as enzymes involved in ubiquitination, like UBC13, but we were surprised to find that deletion of a ribosomal gene, RPL12A, resulted in robust restoration of F670del mediated oligomycin resistance. Moreover, RPL12 deletion emerged as a hit from the screens performed on the premature termination variants. Therefore, RPL12 disruption appears to rescue functional expression of the F508del, W1282X, and G542X CFTR equivalents in yeast. I'd like to note that other ribosomal proteins emerged as hits, and we are following up on those leads as well, but due to time constraints today, I can only discuss the RPL12 story. To briefly introduce RPL12, it is a component of the ribosomal 60S subunit and forms the base of a flexible structure called the P-stalk, which primarily interacts with elongation factors during translation. RPL12 is also a constituent of the GTPase-associated center, or GAC, which can interface with any GTP-bound translation factor. Based on the literature, RPL12 has an established role during elongation through its interactions with the elongation factor 2, but due to its location in the GAC, it's likely that RPL12 plays other roles in different aspects of translation as well. So, taken together, our first aim was to determine whether RPL12 suppression improves delta F CFTR biogenesis in human-derived airway cells. Then, based on the genetic interactions identified from the yeast delta F screen, this suggests that there should be conservation between the yeast and the human airway epithelium, at least so insofar as CFTR processing is concerned. So here we evaluated the effects of siRNA-mediated suppression of RPL12 in primary human bronchial epithelia, or HBE, that were collected from an F508del homozygous individual. It's indicated as patient code 21 there. And this was following pulmonary transplant. These cells are compared to wild-type HBE from a healthy donor that's annotated as patient code 10. So to first orient you to a normal Western blot of CFTR, band C, or that opened arrowhead, indicates the fully glycosylated mature form of the protein, whereas band B, indicated by that closed arrowhead, shows the ER-retained immature glycoform. As you can see for wild type, most of the protein is band C, but for delta F, only band B is present. When we treat delta F with a CFTR corrector like VX-809, which was mentioned earlier as part of an FDA-approved combination therapy, we can see that there is maturation as evidenced by the appearance of band C. And in addition, the vehicle control for VX-809, which is DMSO, does not seem to have an effect. In addition, the RPL12 knockdown appears to achieve a level of F508del rescue that's similar to VX-809. And when these two interventions are combined, there's clear additivity. A non-specific siRNA is also utilized, and it's annotated as the NS that you see there. And it's a negative control for the RPL12 siRNA. The cumulative results from these experiments are quantified on the right, showing band C in the medium gray, band B in the black, and RPL12 in light gray. And I'd like to point out that the degree of RPL12 suppression that's required to rescue delta F in this fashion is modest. RPL12 is only reduced by approximately 30%. So next we wanted to assess whether CFTR functional studies recapitulated the Western blot results. So we utilized a well-established Using chamber protocol to quantify CFTR-mediated transepithelial ion transport. This is a typical tracing that you see here with wild-type CFTR short-circuit currents shown in black. Following sequential addition of CFTR agonists such as forskolin, which is the FSK, as well as genistein, which is the GEN, these are the agonists that are going to amplify the resident cell surface associated CFTR activity. Experiments are always arrested by the addition of a CFTR specific inhibitor, INH172, that's shown there at the end of the tracing. And that's just to indicate whether the observed currents are in fact measuring CFTR activity. So in the case of untreated delta F that's shown there in the gray, there's pretty minimal activation observed, but when cells are treated with VX-809 that's shown in the blue, or with RPL12 siRNA shown in red, delta F channel function increases 2 to 3-fold. And when these two interventions are combined, delta F activity reaches levels approximately 50% that of wild type, and that's shown in the yellow. The cumulative results from these experiments are shown there on the right, with forskolin response in black, genistein in the medium gray, and INH172 in light gray. And these results are significant because reaching a level like 50% the activity of wild type is predicted to be in a range of clinical benefit. So for the sake of time again, I will just summarize some additional evidence that we have collected, which shows that RPL12 suppression increases a lot of different folding aspects of delta F assembly. Thus, our next aim was to evaluate the effects of RPL12 depletion on translation kinetics, and shown there. Based on the established interaction between RPL12 and elongation factor 2, we hypothesized that translation is likely reduced by RPL12 knockdown, thereby allowing the ribosome and associated chaperones more time to achieve a native-like conformation of delta F protein. In collaboration with Dr. Zoya Ignatova's laboratory at the University of Hamburg, we utilized ribosome profiling coupled to RNA-seq, otherwise known as RiboSeq, to assess both global and transcript-specific changes in translation dynamics following RPL12 knockdown. Briefly, unprotected mRNA is digested away and only ribosome-protected fragments are submitted for sequencing. So positions where the abundance of ribosome-protected fragments are higher indicate ribosomal stalling. Using immortalized CF bronchial epithelia that stably express delta F CFTR, we found that RPL12 silencing increased levels of ribosome-protected fragments over the start codon, shown there in the black tracing, as compared to the nonspecific siRNA shown in the gray. Thus, this indicates that translation initiation is partially attenuated. Next, we performed ribosome runoff assays to measure changes in elongation rates. Initiation was stopped using Harringtonine, and at various ensuing time points later, elongation was arrested using cyclohexamide. After submitting the ribosome-protected fragments for sequencing, this allowed us to track the speed at which ribosomes run off the mRNA. So this tracing shows the runoff rate for cells that are treated with RPL12 siRNA with the 0 time point annotated in gray and the 90-second time point shown in green. Velocity was calculated by measuring the slope of that green dip. So in other words, from the lowest point of the dip up to where it merges with the 0 time point curve, and then dividing that slope by the seconds of the runoff. We found that elongation was reduced to approximately 1 to 2 amino acids per second as compared to the usual 5 to 6 amino acids per second under normal physiological conditions. To take it a step further, we wanted to determine whether RPL12 silencing reduced rates of initiation and elongation through effects on specific codons. So from the RiboSeq data, we measured the ribosomal A site dwelling occupancy of each codon over the whole transcriptome, and where elevated occupancy levels correlate with reduced rates of translation, again, due to ribosomal stalling over these codons. We found that RPL12 suppression did not exert its effects on low, medium, or high abundance codons in general, but instead significantly increased A site occupancy of GC-rich codons such as GCG and CCG that are shown there at the end of the table on the right. Next, we evaluated A site dwelling occupancy over just the CFTR transcript. The different subdomains are annotated across the top there, and RPL12 siRNA is shown as the black tracing, whereas the nonspecific siRNA is in red. We discovered that RPL12 silencing resulted in significant slowing at specific mutation or specific positions in CFTR, specifically within the TMD1 R domain and NBD2. Thus, it appears that local reduction in ribosome velocity appears as a general mechanism for functional rescue of delta F CFTR. RPL12 knockdown, therefore, slows the rate of initiation and elongation, primarily through effects on GC-rich codons. And what I didn't have time to show you today is that this is also in relation to the fact that these GC-rich codons pair to high abundance tRNAs. So together, this results in distinct changes in the CFTR translational profile. Our next aim was to assess whether RPL12 silencing rescues other CFTR defects, and due to known interactions of the GAC with all GTP-bound translation factors, we hypothesized that RPL12 depletion may represent a general mechanism for rescue of CFTR variants from several subclasses. Moreover, the E-screens suggest that RPL12 is a modifier of CFTR premature truncation codons. We utilized Fisher rat thyroid cells for these studies, which is a cell system commonly utilized in CF drug discovery laboratories, and it's recognized by the FDA as pertinent to label expansion of rare CFTR variants. So for the sake of time, I'm just going to briefly summarize that RPL12 suppression did partially rescue 2 variants that had folding defects similar to delta F, and that's shown here as the G85E, which RPL12 knockdown alone can increase its function to approximately 8% of wild type CFTR. In addition, the A455E variant, its function was enhanced to approximately 20% of wild type when RPL12 knockdown was combined with a corrector, similar to what we saw in the context of the delta F CFTR studies. However, the P67L variant was not affected by RPL12 suppression. So it appears that RPL12 knockdown can rescue some, but not all CFTR variants from different subclasses. But interestingly, we did want to evaluate the W1282X variant, again, because of the previous C studies that were conducted. So here I'm showing RPL12 knockdown in the primary human nasal epithelia that are homozygous for W1282X CFTR. A number of different small molecule interventions didn't seem to enhance the activity that's shown there by the first few columns. However, RPL12 knockdown by itself increased W1282X functional expression up to approximately 16% of wild type. So this is significant because no other small molecule intervention has been shown to improve W1282X activity to this degree. So again, it appears that RPL12 can rescue some, but not all CFTR defects. And furthermore, it's likely that different mechanisms are responsible for RPL12 mediated rescue, depending on the nature of the CFTR defect, as well as the quality control pathways that are involved in its processing. So lastly, our future directions are going to include characterizing the mechanisms by which silencing RPL12 and other ribosomal proteins identified from the yeast screens overcome CFTR premature truncation codons. This could be by inducing read-through and or inhibiting nonsense-mediated decay or other mRNA surveillance pathways. We will also evaluate whether RPL12 depletion rescues the CF phenotype in humanized delta F CFTR, W1282X CFTR, or G542X CFTR mouse models. This is a collaboration with Craig Hodges at Case Western. And we are going to be using an RPL12 heterozygous animal that we generated. Here shown, where these mice have half their levels of RPL12 suppressed, but they are all expressing wild type CFTR. And so, for example, we found in the pancreas, colon, and ileum that half the RPL12 reduced results in enhanced maturation of even wild type CFTR. So we hypothesize that it may have effects on these other mutant forms of the protein as well. And lastly, we are working on validating antisense oligonucleotides that have been generated against RPL12 for preclinical evaluation in CF model systems. And this is that active collaboration I mentioned with Ionis Pharmaceuticals. So I'd like to acknowledge that this project has been highly collaborative and multidisciplinary. Several investigators who have worked with us over the years, most notably my co-mentors, Eric Sorscher and Zoya Ignatova. I'd also like to acknowledge our funding sources from the NIH, Burroughs Wellcome Fund, and National CF Foundation. So thank you again for the opportunity to present today, and I'm happy to take any questions.
Thank you, Dr. Oliver. We're a little tight on time, so I'll go ahead and entertain one question for Dr. Oliver.
I'll be happy to ask a question. Catherine, that's really elegant work. Congratulations. So you're knocking down expression of the ribosomal protein with antisense molecules. Does that have a potential therapeutic application, or would you be seeking small molecules that were inhibitory for the protein?
The short answer is yes and yes. We can use small interfering RNAs. That's the collaboration with Ionis is actually what that is working towards. They're antisense oligonucleotides that have a little bit different of a biochemistry and a structure to them compared to siRNA, and so they're more stable in vivo. They are more effective. They last longer. They're actually already approved for preclinical evaluation as well. So if we were to get promising in vitro and in vivo results, we could move straight into a clinical trial with those oligos. But we also don't want to put all of our eggs in one basket, so to speak. So we are also working on screening for anti-RPL12 small molecules.
Okay, thank you, Dr. Oliver. So in the interest of time, we'll move on to our final oral presentation of the day.
Yes, which will be given by Dr. Loretta Reyes. Loretta is assistant professor in the Division of Pediatric Nephrology. She also is one of our Atlanta Pediatric Scholars, K-12 scholars. She's been at Emory on the faculty now for 2 years, and prior to that, she was a fellow in pediatric nephrology. She received her MD from St. George's University School of Medicine and completed her residency at Driscoll Children's Hospital in Corpus Christi, Texas. Her mentors are Claudia Morris, Luann Brown, and Roy Sutliff, and her research focus is on cardiovascular factors, arginine dysregulation, and metabolomic profiling in chronic kidney disease. So Loretta, take it away.
Thank you, everyone. And I'd like to thank the organizers of today's session for inviting me to present my work. I will be presenting arginine dysregulation and correlations with cardiac remodeling in mice with chronic kidney disease. I have no conflicts to disclose. As some background, chronic kidney disease is defined as a glomerular filtration rate of less than 60 mL per minute per 1.73 meters squared for greater than 3 months, where GFR is a measure of the clearance capability of the kidney. There are 5 stages of chronic kidney disease, and kidney function decreases during each stage. End-stage renal disease or kidney failure is defined by a GFR less than 15 and the need for renal replacement therapy in the form of dialysis or a kidney transplant. And patients develop multiple complications with progressively worsening chronic kidney disease. And unfortunately, the risk for mortality increases with increasing CKD stage. The increased mortality seen in CKD occurs due to cardiac disease, and this data was initially presented in 2012, but annual US RDS reports continue to show this trend. In the general population, a cardiac cause for death occurs in less than 5% of cases. However, in children on hemodialysis or peritoneal dialysis, a cardiac etiology accounts for approximately 30% of cases. After kidney transplant, cardiac disease still accounts for approximately 20% of deaths. Endothelial dysfunction likely plays a significant role in the pathogenesis of cardiovascular disease in CKD. Nitric oxide is essential for vascular homeostasis, and endothelial dysfunction occurs when there is reduced nitric oxide synthesis or bioavailability. The precursor for nitric oxide synthesis is arginine, which is endogenously produced in the kidneys. CKD is a nitric oxide deficiency state, and in kidney dysfunction, there may be reduced arginine synthesis, diversion of arginine into other metabolic pathways, and inhibition of the nitric oxide synthase enzyme. From this pathway, we can also see that the availability of arginine can be affected by its catabolism products and its precursors, and this allowed for the construct of global arginine bioavailability ratio, or GABR, which has been used as a measure of arginine's true bioavailability. Global arginine bioavailability ratio has been studied in different patient populations with endothelial dysfunction that's contributing to cardiovascular complications. My mentor, Dr. Claudia Morris, identified increased mortality in patients with sickle cell disease and low global arginine bioavailability ratio, as highlighted by the red arrow. Similarly, this adult study in stable coronary artery disease patients demonstrated lower global arginine bioavailability quartiles, again highlighted by the red arrow, with increased risk of non-fatal, was associated with increased risk of non-fatal myocardial infarction, non-fatal stroke, or death within a 3-year follow-up period. And these patients all had traditional cardiovascular risk factors, which include diabetes, hypertension, and dyslipidemia, similar risk factors to those seen in patients with CKD, but in these patients, the renal function was intact. Interestingly, global arginine bioavailability ratio has not yet been studied in the CKD population. With this background, we decided to investigate arginine metabolism in mice with CKD and sought to identify any correlations with cardiovascular complications. CKD was induced by the remnant kidney model. This is briefly where young male 129 SVJ mice were randomized to undergo 5/6 nephrectomy via a 2-stage surgical procedure. For the first surgery, 2/3 of the left kidney was removed, and 1 week later, the right kidney was removed via a second surgery. Sham controls underwent surgery to expose their kidneys, but tissue was not removed. The completion of the second surgery is considered the onset of CKD. The CKD and sham controls were followed for 16 weeks, and plasma, tail cuff blood pressures, and echocardiograms were obtained at 4, 8, and 16 weeks of CKD. The plasma was analyzed via mass spec for arginine, ornithine, citrulline, and ADMA. Colorimetric assay was used to measure arginase activity. And then echocardiographic measures obtained included LV mass, relative wall thickness, E to A ratio, MPI, and global longitudinal strain. In our first series of experiments, we analyzed the metabolites of the arginine metabolic pathway and correlated them with cardiovascular measures. The bottom row highlights that global arginine bioavailability ratio demonstrated a non-significant reduction at both 8 weeks and 16 weeks of CKD. The first row, however, showed that ADMA was significantly increased at 16 weeks of CKD compared to controls. And similarly, the second row shows that arginase activity was significantly increased at 16 weeks of CKD versus controls. We next correlated global arginine bioavailability ratio with echocardiograms, and in mice with CKD, lower global arginine bioavailability ratio correlated with a higher relative wall thickness, which is a measure of left ventricular hypertrophy. Additionally, the lower global arginine bioavailability ratio also correlated with a lower E to A ratio, a measure of diastolic dysfunction. As mentioned previously, arginase activity was increased in CKD mice compared to controls at 16 weeks. When we specifically compared CKD mice at both 8 and 16 weeks, we saw that arginase activity was higher in the CKD mice at 16 weeks compared to at 8 weeks. We then correlated arginase activity with global longitudinal strain and showed that arginase activity correlated with a lower global longitudinal strain, which is a measure of systolic function. In our second series of experiments, we investigated the effects of arginine supplementation on the plasma and echocardiographic findings in our CKD and control mice. Once again, CKD was established via the stage 5/6 nephrectomy, and mice received a diet supplemented with arginine or alanine as a nitrogenous control for the duration of the experiment. The experiment was concluded after 12 weeks of supplementation, at which time plasma, blood pressure, and echocardiograms were obtained. After 12 weeks of arginine supplementation, relative wall thickness, a measure of left ventricular hypertrophy, was improved in arginine supplemented CKD mice compared to alanine supplemented CKD mice. Additionally, myocardial performance index, or MPI, which is a measure of systolic function, was improved in arginine supplemented CKD mice compared to alanine supplemented CKD mice. And after 12 weeks of arginine supplementation, arginase activity was also seen to be significantly lower in arginine supplemented CKD mice compared to controls, and in arginine supplemented CKD mice compared to alanine supplemented CKD mice. So in conclusion, we found that arginase activity was significantly increased in mice with CKD. This occurred after a longer duration of CKD. The implication of this is that this may have clinical relevance in the assessment of children and young adults with more advanced CKD and end-stage kidney disease. Dysregulated arginine metabolism correlated with myocardial dysfunction in mice with CKD, and the mechanisms by which this dysregulated metabolism contribute to myocardial dysfunction is being further elucidated to help identify targeted therapies. Arginine supplementation improved left ventricular hypertrophy and myocardial performance index in mice with CKD, and similarly, arginine supplementation resulted in decreased arginase activity in mice with CKD. The implications of these findings will also be investigated further. Future studies. So the pilot data generated in these experiments has raised several questions, which we're currently investigating. Currently on my K-12, I am assessing the correlations between arginine metabolism and endothelial dysfunction in mice with CKD, since endothelial dysfunction has been proposed to be the mechanism for cardiovascular complications in many disease states. Secondly, the advances in the field of metabolomics has identified metabolomic footprints in several disease states. The dysregulation in arginine metabolism seen in these pilot studies therefore warrants a more thorough assessment of the arginine metabolome to determine potential therapeutic targets to ameliorate the cardiovascular complications seen in children and young adults with CKD. At this time, I'd like to thank my past mentors, Claudia Morris, Pam Winterberry, and Luann Brown, as well as my current mentor team, which now includes Dr. Roy Sepleff. I'd like to thank the members of the Pediatric Biomarkers, Biostatistics, and Animal Physiology cores for their assistance with these projects. And I will take any questions at this time.
Thank you, Dr. Reyes. That was an excellent talk. And as someone who's watched you progress in your career, I can say that you've done an excellent job locating and finding great mentors to really complement and round out your complete mentor portfolio, as it were. So people may want to follow up with you on that account as well. But we'll go ahead now and take any questions. Would any of the judges like to pose a question? Dr. Trudy?
Yeah, this is Anne. That was a great presentation, very clearly understandable from someone who is not at all in that field. One thing you mentioned at the end kind of perked my ears up as well, the idea of the endothelial dysfunction. You know, as everything seems to relate to COVID these days, that's a kind of a hot topic in that area as well. And I was just wondering kind of what are your plans to, what are the markers that you use to evaluate that, and how do you plan to do those experiments?
Sure. So right now I'm looking at it in a mouse model of CKD. So essentially we're looking at the aortas and looking at relaxation responses. But for clinical relevance, we're also able to look at the pulse rate velocity, CIMT. So we are able to look at clinical measures of endothelial dysfunction in the human population. So that will be sorry, that will be something that we need to look at, that we can look at in patients with CKD, patients who have other endothelial dysfunction, comorbidities who are also impacted more significantly with COVID.
Thanks.
Other questions? Looks like we have time for one more. If anyone has a burning question they'd like to ask, might be somebody on your phone. We don't know. Okay, so with that, I think we'll conclude. Thank you to all our fantastic oral presenters. The next part of our presentation, Dr. Joyner, would you like to walk people through the next part and what comes
Sure.
next?
So we will now go back into the Zoom rooms to begin the networking and poster session portion of the program for basic science posters. So we encourage you to visit with the poster presenters by clicking on their individual Zoom links in the program for each presenter. So if you go to the program with the name of each presenter, you'll find a Zoom link. And then you can go from Zoom room to Zoom room, and of course you have to leave one room before you go to the next one. So we'll break now and resume the program at 4 o'clock. Please come back at that point for the presentation of the basic oral and poster award winners, and for the wrap up of the program. So with that, we'll stop this part and move on to the Zoom rooms.
See you all at 4.
Organizing this event, including Dr. Miriam Voss and Dr. Claudia Morris, Dr. Lazaros Kachillis, Dr. Jocelyn Greenwald, and Dr. Holly Gooding, as well as Barbara Kilburn, Carol Flowers, and Kim Houghton, who really did the incredible work of putting all this together. And you know, I will say too that this was a new experience for us, running a virtual meeting, and it's a little bit of a trial run for our hosting of the NICHD K-12 National Program meeting, which will happen in October, and which will also be a virtual meeting somewhat similar in format to this one. So this has been a great trial run. And now I'll turn it over to Stacy to have, well, first let me ask if Dr. Jane wants to have any final words, and then we'll let Stacy wrap it up.
No final words. Just, Clint, I want to thank all of you for the hard work that went into putting this really amazing meeting. Stacy, I must admit to you that I felt that the program got enriched by including the clinical presentations. Last year, or the 2 previous years, as most of you know, we had mostly basic science presentations, given that this is a K-12 oriented program. But I do think that getting young people together, and in particular, your choice of Ed Rudy as a speaker was just, I mean, the whole program was amazing. So thank you very much.
I thank you so much, Lucky, for those really wise comments. I couldn't agree more. You know, this was something new for us. We tried to take some best practices from other conferences that we'd heard about that were good. At the same time, we hope you enjoyed today. We hope you got some great nuggets and interactions and met some new people. But we are really interested in getting your feedback. And to that end, we'll be sending you a survey, and we just really hope that you'll respond with sort of all kinds of feedback, constructive feedback, as well as any highlights that you thought about today. I think that, you know, the one thing that I miss most about being in person is the networking opportunities, the ability to be face-to-face with someone and reading body language and really just expressing our enthusiasm towards each other's research. And so if anyone has been to any conferences that you think have done that in a really effective way in the virtual setting, please use the survey to let us know. because we do intend to do more of these. The K Club's coming up this fall, and we really want to make sure that it continues to be that enriching experience. We're not going to do food delivery to your homes, but hopefully you'll join us and grab your lunch and sidle up to your home desk and still participate in the K Club. And we do intend to keep those going. So I just want to, in closing, just thank everyone again for this great showing today throughout the day. And once again, Barbara will be sending out the survey and really want to get your feedback and what we could do better and differently next time. So thank you, everyone, and enjoy the rest of your Tuesday.
And Stacy, thanks again to you. Clint, I must say that you are very lucky to have somebody like Stacy Heilman to help with not just this type of a program, but I think Stacy has uniquely focused on helping foster research activity amongst younger people. And I think the platform, K-12 is just the tip of that iceberg that we're trying to build. Ultimately, the hope is that there should be a lot more people in the department, in the Pediatric Institute doing this kind of work. And we can't thank Stacy enough. And of course, as Stacy, you mentioned, there's a team behind you, Barbara, Kim, and I'm on board. Carol, exactly. Carol, I saw Carol in the virtual rooms as well. So thanks to all of you on the team and for this wonderful show today.
Thank you, Lucky. Appreciate that. Hope everyone has a wonderful day, and we'll see you soon.
Good afternoon, everybody.